Hello all. So today let us discuss with the experiments related to photosynthesis. So we have some experiments what we have to memorize. So these are the activities or experiments related to photosynthesis. So you remember the equation whenever you think of photosynthesis, just recollect your equation of photosynthesis, which is 6CO2 plus 12H2O. In the presence of light and chlorophyll, it is giving rise to glucose, which is C6H12O6 plus 6H2O and 6 molecules of oxygen is getting released. So this particular equation always try to memorize. So coming to the experiments part, so what are these experiments? In these experiments, we will be actually discussing about the components, that is the four raw materials that are there: carbon dioxide, water, sunlight, chlorophyll. Are they really essential for photosynthesis or not? We will work out as experiments and see whether these are really needed for photosynthesis or not. Also, we will discuss with experiments related to production of glucose and oxygen. Really glucose is getting produced or not? You can check out through the experiment. And also really oxygen is getting released or not? We can check through the experiment. So all these experiments we will be discussing. Okay. So coming to the first test or the first experiment, that is called the starch test. What it is? It is called the starch test. So what is the starch test all about? See, children, first we have to learn, a test that is called starch test. You know that by the process of photosynthesis, glucose is getting produced, and this glucose throughout the day photosynthesis takes place, and it will be getting stored in the form of starch in the leaves, starch in the plant parts. So first test what we will do is we will test whether really starch is there in the plant because of photosynthesis or not. So for which you have to first understand starch test before understanding any other experiment. So let us discuss with the starch test. So what is the starch test? So you take a potted plant. What we will do? We take a potted plant. That is a plant which is present in the pot is what is called the potted plant. So keep it in sunlight for photosynthesis to take place for 2 to 4 hours. Okay. So after the period of 2 to 4 hours, you take out a leaf from that and then you put it in a test tube. You will put it in a test tube containing methylated spirit or alcohol. Methylated spirit or alcohol is the substance that is present. So this one, the alcohol or methylated spirit, why we are using is, this is a decolorizing agent. Leaves as such will have intense green color. So because of which the color changes that will happen in any particular test are not visible. So you will be first dipping it in alcohol or methylated spirit, which is your methyl alcohol, and then you will boil it so that the color of the leaf gets reduced, and then you can carry out the further test to see visible reactions, So here we have taken the leaf after the period of photosynthesis, we have put it in a test tube containing alcohol. So I hope all of you know how a test tube looks like. So this is a test tube actually, and so alcohol will be colorless like this, and you will place your leaf inside this leaf inside the alcohol like this, and then we will carry out our experiment. Okay. So next what will happen is, so you are taking the test tube and we are keeping it in the dish which is containing water in it. So this is what is called water bath. So you take a beaker or you take a dish or small vessel and pour water in it, and first we will heat the water. Okay, and then in that we will keep our test tube containing the leaf with the support of a tongs or a test tube holder. You will hold your test tube and keep it in the water bath. Okay, for a period of some 10 minutes you can keep inside that. What is the purpose of this water bath? Is water bath provides uniform temperature for heating. Otherwise, what will happen if you are keeping your test tube directly on the flame? Okay, the test tube may burst, because it is made up of glass material. And one more problem is alcohol that is placed inside, it has got low boiling point of about 70 degrees centigrade. So that will bump out. It will be jumping out when you heat the substance directly on the flame. So that's why, to avoid all the problems, we are using a water bath in which you keep the test tube with the help of the test tube holder, and after heating for a few minutes, you take out the leaf, take it in a small petri dish. Okay, so in our lab we have a small glass container which will be called the petri dish. We can place it in a petri dish, it is called as. So in this you keep, and then you add few drops of iodine solution. So iodine reacts with starch and will give a blue black color. So that is the principle of this test. Anywhere you take, children, if you remember in your 6th class, okay, we have done an experiment in science wherein your teacher might have taken some foodstuff from your carriage and she might have done the iodine test, if you remember, the protein test, iodine test and all. So wherein when iodine is added to any carbohydrate material, what will happen? It will change to blue black color, if you remember. So the same principle applies here. So starch test you will have. So once you add your iodine solution, the leaf becomes blue black in color. So this is the test for the presence of starch. If there is a color change, it indicates that your leaf is having starch in it. If there is no color change, then okay, your result is negative, your starch test will be negative. When it will be negative? If you keep your plant in shade totally, not exposing it to sunlight or any light at all, in that case when you perform your test, starch test, then it will be negative, okay, because there is no photosynthesis in such plant. Okay, so like this we can check out, okay, whether really starch is getting produced by the leaves or not. Okay, this is starch test. So you have to remember this starch test forever, because in all other experiments we will just refer okay, the end reaction as do the starch test, wherein you have to remember that the entire test will be performed. Okay, so this is the first test, starch test. Hope you understood. Take a potted plant, take a leaf out after the period of photosynthesis, then you will keep it in a test tube containing alcohol in it, and we will boil it in a water bath. Take the leaf out and you add iodine solution. After adding iodine solution, if it becomes blue-black color, then it indicates presence of starch. Okay, coming to the next experiment. Okay, this is called the Moll's half-leaf experiment. What it is? It is called the Moll's half-leaf experiment, or it is the test for carbon dioxide. So now this is a test to see whether really carbon dioxide is needed for photosynthesis or not. So this was actually devised by Moll. So the experiment was performed by Moll. So it is called Moll's half-leaf experiment, because in this half-leaf will be checked out for presence of okay, starch, that is whether photosynthesis has taken place or not. We will check in half portion of the leaf. So that's why this is called as Moll's half-leaf experiment. Okay, so coming to the experiment, you take a potted plant and then keep it in the sun for 2 to 4 hours. Okay, and then okay, what we will do, one of the leaf you take and then bend it and put it into a glass jar. Put it into a wide mouth glass jar as I have drawn here. So in this glass jar, what we will do is we will put a substance called as potassium hydroxide, a chemical called as potassium hydroxide in one corner. So this potassium hydroxide, the function is very, very important. What it will do, it will absorb of all the carbon dioxide that is present within the bottle. That is the function of potassium hydroxide. So now what you are doing, you have taken one of the leaf and then you are seeing that half of the leaf is going inside the bottle, as you can see here, and half is present outside. So in order to facilitate this particular arrangement, we have a cork which is called a split cork. Split corks are available. So what is split cork? A cork will be there with a small slit will be there in the middle. So through that slit actually we will insert our leaf half portion into the bottle. And then so what is the status? What is the environment inside the bottle? Yes, it is not having carbon dioxide inside. So now you are keeping it like that, and again you keep it in sunlight. Okay, allow the photosynthesis to be completed. Then you take this leaf out. You cut the leaf. We have cut the leaf out, and then we perform the starch test. So what will happen, children? Any guesses? Yes. So the portion of the leaf that is present inside the bottle, this was the portion of the leaf which was present inside the bottle. It is not having carbon dioxide. So no photosynthesis, no presence of starch. So what is the final result? There will not be any blue-black color for the portion of the leaf which was placed inside the bottle. For the portion of the leaf which was outside the bottle, the starch test will be positive. Starch test will be positive. That is, it will become blue-black in color because it is having all the components. Carbon dioxide is there for the outer side portion. Water, carbon dioxide, sunlight, chlorophyll. All 4 factors are there. Whereas inside the bottle, there is no carbon dioxide. So because of this, actually, okay, the portion that is present inside the bottle will be not turning blue-black in color after performing the starch test. Okay, so hope you understood. Just we take a plant and then insert half leaf into a bottle which is containing potassium hydroxide pellets, which will absorb of all the carbon dioxide inside. So after allowing it for photosynthesis, when you check the result, the portion of the leaf which was there inside the bottle, it will not turn blue-black in color, whereas outside one will turn blue-black in color. So this indicates that the presence of carbon dioxide is really essential for photosynthesis. Coming to the next experiment, importance of water. So water is important for any living organism to survive. Even plants need water. So if you are not putting water to the plant, okay, the plant parts will wilt and the plant will die. So there is no question of photosynthesis at all for the plant. So the plants will obviously die without water. So that is the indication, okay, that they are not able to produce. Why they die? Because they cannot produce any food. When they cannot produce any food, they cannot survive and hence they die. So this is the test. Next we have is experiment, okay, to prove that sunlight is essential for photosynthesis. Okay, whether really sunlight is essential or not for photosynthesis, you can do a small experiment. So what we will do, we will take a potted plant again. Okay, you take a potted plant. And what we will do, to one of the leaf, you tie a black paper. We take a black paper with the help of gum clips, which will be used to secure your papers. With the help of those, we will tag this black paper there to one of the leaf. You cut a small portion, okay, any shape, whatever you want, in the middle of the black paper also, and then tie that, and then keep this set up in the sunlight for 2 to 4 hours. So after the period of time, what will happen? You can cut the leaf and then you perform the starch test. We perform the starch test. So what you think will happen? Okay, yes. So the portion of the leaf which is getting sunlight, that is this portion, the other portion, and also the portion that is cut in the middle. So all these portions are receiving sunlight and also the other factors that are essential for photosynthesis. So all these parts will become blue-black except the portion which was blocked with the paper. So here you can see the result. So this portion has become blue-black, even this one, and the cut portion has also become blue-black in color. But this portion remains green in color, okay, because there is no sunlight. We have blocked the sunlight there. So this indicates that when there is no sunlight, okay, in one particular part, when you are blocking the sunlight, okay, then there cannot be photosynthesis that can happen, and the end result after starch test would be that portion remains green in color only. So this is a simple test to indicate sunlight is essential for photosynthesis. Children, there can be a question, is that sunlight only will help in photosynthesis? Sometimes even artificial light also will facilitate for photosynthesis. They do what is called tissue culture. If you remember that Bale Bale Magadi Boy movie has come. In that movie, actually, that first shot, in the first shot, hero Nani will be doing something in a lab. Okay, so if you have not watched, you see the first scene. So actually some plants, what I mean to say is, some plants are grown in the lab also artificially. That time artificial source of light will also work out for photosynthesis. It is not that always sunlight has to be present. Okay, so coming to the other test, we will also learn about the tests, okay, to prove that chlorophyll is really essential for photosynthesis, and also we will see an experiment to show really oxygen is getting released during the photosynthesis. So let us move on and find out with the other experiments how it will happen.